So Cynthia, how do you use the collection in your work?
And just a couple years ago, we did work, and this is an old picture from the 1930s of Schenley Plaza, which is right outside the museum. During that time, they had planted, back in the late 1800s, 200 trees. They're the sycamore trees. Over the 100 years in which they were planted, approximately 100 of them died. And several years ago, Phil Gruszka from the Parks Conservancy came to me and said, "Can you tell me about the genetic makeup of the existing trees in Schenley Plaza, but also tell me about the genetic makeup of the trees that we're going to plant?" Because he was very conscientious and he wanted to make sure that he was planting nice, diverse, genetically makeup of the trees from the nursery stocks. What we did is we actually got a bunch of different leaf samples, and this is a little diagram just kind of showing you. We got different leaf samples from out there in the plaza area and also from the nurseries. So we got the complete DNA out and we cut it up. We cut them with special scissors. And with those scissors, we had all these little tiny fragments and we lined them up from the smallest to the biggest.
I have to ask you, how are the scissors special?
Because they're called restriction enzymes, and they're special because they cut DNA. And they cut DNA in the exact precise spot that you ask them to.
So they're not physical scissors?
They're not physical scissors,
Gotcha.
but we constantly refer to it in that way because it gives people a good idea, an image in their mind to think about with what we're doing with these techniques. So then we line these up from the smallest to the biggest, and this is just one plant. And we did that with all the plants. And then their banding pattern will show us the differences between these trees.
Okay.
And when we actually put them together, we can say, these two are closely together, these two are closely together, this one and this one is more farther apart. And so it will tell us something about their genetic makeup, their history, their relationship to one another. And what we found, amazingly enough, which this may be difficult to see, is that these long branches indicate nice genetic diversity, lots of genetic diversity. All the Schenley Plaza trees pretty much had tons of genetic diversity, where the stuff that came from the nurseries had long branches out to them, meaning that these were all the same, and then they diverged right at the end, meaning that they didn't have very much genetic diversity. I then said to myself, what is going on here? I thought perhaps Phil, my colleague, got confused and collected the same leaves. So then we went to another nursery. We found the exact same pattern in the other nursery. So then we started looking into it and we realized 100 years ago they didn't have these modern techniques to actually take cuttings and to grow the plants up. They use seeds, and with seeds you have basically an egg and a sperm coming together and you have genetic diversity. But nowadays they use cuttings from this genetic cultivar that they think is more superior. Doing this is decreasing our urban diversity. And there are millions of dollars being spent on our parks nowadays here in Pennsylvania, but also all the way throughout the country to green things up, to make the environment better, reduce our carbon footprint, make the temperatures better by having plants cool the environment. And no one's doing any genetic studies at all and looking at what they're planting. So they may be spending millions of dollars on planting clones, which could be setting this up for a disaster.
What kind of disaster?
Well, if we plant something out there that's all exactly the same, and another disease comes through like 15 or 20 years ago, 15 or 20 years ago, these trees had, some genetic diversity. They were different, so they actually had more of a resistance to the disease. Now, if we do the same thing with the plants that are being planted nowadays, the disease is more readily to go through and wipe out each one of those plants. And what we have done is we have given some of the cuttings of the ones from Schenley Plaza to nurseries so they can start growing those. And the reason why we did this is because we know they're genetically diverse. We know they've survived 100 years and they've actually been through this disease more than once, more than several years than this cultivar did. And we know that it's a good genetic stock and there's something that the nurseries can use to use these cuttings once again, but to have an increased source for their genetic diversity, not just one or two plants.
Cool. And that'll put an end to Franken trees.
Hopefully.
Hopefully.